Good afternoon. My name is Lynn Young, and I'm a professor at the UBC Graduate School of Journalism and co-founder of The Conversation Canada. Welcome, everyone. I'd like to start off by acknowledging that the land that we're bringing this webinar to you on in Vancouver is the traditional, ancestral, and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh peoples. A sense of place and belonging are an integral part of daily life, especially in the current climate, and we want to acknowledge this privilege before we begin. On behalf of Alumni UBC and the University of British Columbia, I'd like to extend a warm welcome to everybody who's joining us today for the webinar, whether you're viewing from the Lower Mainland, across Canada, or elsewhere in the world. We're pleased to be able to present this online webinar during the unprecedented time that we're all facing, and this new reality that we're all trying to get to know and manage. Scientists from around the world, including Canada and at UBC, are working tirelessly on the development and testing of new treatments and vaccines to help contain the current outbreak of COVID-19. Among them is UBC's Dr. Josef Penninger, who's with us today. He's the senior scientist leading an international research team that has found a trial drug which effectively blocks the cellular door that the novel coronavirus uses to infect us. The team's findings hold promise for a treatment capable of stopping early infection of COVID-19. We're lucky to have Dr. Penninger here with us today, tuning in from Vienna, Austria. I'd like to welcome him and thank him for his time during a period when his #1 priority is research on this important area. Josef Penninger is a professor and Canada 150 Chair in Functional Genetics and the Director of the Life Sciences Institute at UBC since December 2018. Prior to this, he was the Scientific and Founding Director of IMBA, the Institute of Molecular Biotechnology and Austrian Academy of Sciences in Vienna, Austria. And before that, he worked as lead researcher at the Amgen Research Institute in Toronto. Welcome, Dr. Penninger, and thanks for joining us today.
Good evening from Vienna. So it's a pleasure to be with you. Unfortunately, I cannot be in Vancouver because I got stuck before the borders came down in Europe, and so I will join you from Vienna, from my library, which I still have here.
Well, it's great to have you from your library and here. I have some questions that I'm going to start off asking you first for the first half of the conversation. And for the second half, I'd like to address the webinar audience. I'll be taking questions from participants via Slido, which is our online audience engagement platform. You'll have received instructions when you registered for the webinar, and many of you have already inputted questions, so thank you. But as a quick reminder, to input your questions, please go to slido.com and use our event code treatment to sign in. And we're looking forward to some interesting and good questions for the latter half of this conversation. So to start off our discussion with Dr. Penninger, I'd like to hear a bit more about Josef and his team's latest finding. And if you could just maybe tell us a bit more about it as we start.
Oh, yeah, sure. So actually the last 2 months, I put together an international team. So from us at UBC, people from Stockholm in Sweden, because they have a very high security laboratory. And 2 months ago, we already isolated the virus and we kept it in Stockholm under very secure conditions. A team from Barcelona, some biotechs, also a biotech from Vancouver. And basically within 4 or 5 weeks, we all got together and asked the question if we can develop human models for infection. So we basically engineered human tissues from stem cells. So that's what I do. I'm a tissue engineer and want to figure out how diseases happen and what we can do about this. And so we basically engineered human kidneys, we engineered human blood vessels. Having the virus in Stockholm, we infected them, and we basically could show that these tissues can be directly infected, which is very important to understand the pathology and the disease of COVID-19. And at the same time, based on my work, there's actually a biotech in Europe which developed a medicine which is going either this week or early next week into COVID-19 patients for a clinical trial. And so I had access to this medicine being developed for many years now. And we actually tested this, how it affects the virus. And basically it reduced the virus by a factor of 1,000 to 5,000 times. So that's what we found. The reason why this works is because this medicine actually is the gateway or controls the gateway of the virus to infect us. And by doing so, we basically trick the virus and it cannot infect us anymore.
That's fantastic, and what great timing. I mean, I know this comes from a long historical research program that you've been working on, and I know that you're in part funded by the Canadian federal government through emergency funding focused on accelerating the development, testing, and implementation of COVID-19 treatment. And I assume that this is also building on your work at the University of Toronto and the Institute of Molecular Biology in Vienna. So when you and your colleagues first identified ACE2, can you tell us a bit about the history coming into this and your early findings and how that linked to SARS and what sort of, can you start from the beginning basically and walk us through what happened?
Oh, yeah, absolutely. So I was a young investigator in Toronto at the University of Toronto. So I'm a medical doctor and a trained immunologist. And in 1999, we actually studied the development of hearts in insects. So we wondered, we wanted to figure out the genetics of heart development in insects, in a particular insect called the Drosophila fruit fly. And then we actually realized and hit the gene, which we could not really explain because it did not exist yet in mammals or in humans. And actually a postdoc of mine, Mike Krakauer, a Toronto kid, a Canadian kid, cloned the gene, which we then called ACE2, because it was very similar to a sibling, which is called ACE, which we knew for many years before. ACE stands for angiotensin converting enzyme. And since it's the second one, we called it ACE2. So this is where the name came from. It was very curious because nobody anticipated something like this. So we engineered the first human, we engineered animals which basically lacked ACE2. And then we could, for the first time, show that ACE2 is protecting our hearts from heart failure, or the hearts better from mice engineered to lack this gene. So this is where we came in. What we found very early on was a negative regulator of a system we had been knowing, and this system actually triggers heart disease, kidney disease, liver disease. This system triggers hypertension. So this is where the ACE inhibitors come in. This is a raging debate now if ACE inhibitors or angiotensin converting enzyme inhibitors would be good for COVID or not. So this is all based on our research back 18 years ago, which we published in a paper in Nature. So then we realized ACE2 was actually expressed in the lungs. And we had no idea what's actually happening. And so I reached out to an intensive care physician, Art Slatsky, who is still in Toronto and an amazing MD, and he is helping us a lot now. So a big kudos to Art. And I sent one of my students to Art's lab. And actually what happened, and normally I don't tell the story, actually my student fell in love with the person who worked in Art's lab. And this person had developed models for lung failure and lung injury. And so this is how we actually ended up working on lung failure and lung injury, and figured out ACE2 protects lungs from severe damage, something called ARDS, acute respiratory distress syndrome. This is basically what kills patients with COVID-19. So we had been working on this. Then all of a sudden, there was this notion that ACE2 is actually the entry gate for this first SARS virus to infect us. And since we had actually the first mutant animals, we sent our mice immediately to China, because in China they had the virus, the SARS virus. We infected the animals, and sure enough, our animals didn't get any infection and were totally healthy. So basically we cloned the gene ACE2, which is the entry point for the virus. We found out what it's doing, protecting tissues. And thirdly, it turned out it's the essential entry gate in the body, which is important for the virus to actually infect us. So we had done all of this and basically, figured out why this is all happening. And the reason why the SARS virus became such a killer virus compared to other coronaviruses out there is because the SARS virus hit a molecule called ACE2, which actually protects us. It's like HIV, HIV infects by binding to an entry gate called CD4. CD4 disappears, and that's why people lose immunity and get infections. So in the SARS disease, this became a killer because it uses the molecule called ACE2 as an entry gate. And by doing so, it takes ACE2 away and we lose our protection. And this was a molecular explanation why the SARS virus became a killer virus. So, and based on this, we actually developed a medicine with the idea the SARS virus showed us something which could protect our tissues. And then, of course, SARS disappeared. Nobody cared anymore about our discoveries in ACE2. It turns out COVID-19, the virus called SARS coronavirus number 2, uses ACE2 as the entry gate. Basically, everything we had figured out back then in the early days, you can now translate to the infection of this new SARS virus, which causes the disease we are calling now COVID-19. So all of a sudden, we discovered everything. I used to be Mr. ACE2 in the world. Then nobody cared about our work anymore. Now I'm basically at the center of the global storm because nearly every university, every company is working now on this pathway.
That's fascinating, and what an amazing sort of history and story in terms of how it developed and how your knowledge developed in this area. So what's the difference between SARS and the coronavirus now? Like can you tell us a bit about that?
So the entry gate is the same. So the infection using ACE2 is the same. So that's why we actually understand a lot. And I love viruses. I work with viruses. I work with Ebola viruses. We worked with the Spanish flu. So that's part of my life, trying to figure out how they work. So there's nothing to be afraid of viruses because we can understand them. And if we understand it, we can do something about this. The differences are the first SARS virus. And I actually used to work in Toronto at this time, at the Princess Margaret Hospital. So our hospital was more or less under quarantine. So I lived through this. One of our students was infected. But in the first outbreak of the SARS virus, it was very difficult to infect somebody else. So you had to be really sick, cuff somebody in the face, and then you could pass on the infection. So, and of course, by doing so, it was relatively easy for a well-established public healthcare system, like in Canada and of course in China also, to control the infection. And at the end of the day, there were 8,000 people infected, with around 800 people who died. So the fundamental difference with this new virus is, because some of the people who get it are apparently asymptomatic or have not many symptoms. There are data from Iceland, for instance, that 50% of people have the virus and can pass it on to others, have no symptoms. There are data from South Korea where 20% of people are infected, but they are healthy. And of course, if you have a virus which does not make everybody sick and can be relatively easily transmitted, because if I breathe at you and it lives on surfaces, then we have this global pandemic, because this has basically all the ingredients of the global pandemic. So very interesting reports. What would it actually take to have a pandemic? Bacteria don't really qualify much anymore because with antibiotics, which work reasonably well. Fungi, they need their own little climates, so they might not go through the whole world. Malaria is, of course, a big disease we still have to address. So, and the prediction was this virus, which can be, has some incubation time that can already spread, where people are already still healthy and can spread it. And that's why we cannot isolate people. That's why people fly on airplanes, spread it all over the planet. That's what we have been seeing and probably still see. And that's why it's very difficult to contain. The hope, of course, is that it's not as lethal as the first SARS virus, which had 10% lethality of people who got the disease. Also, if I look at the Johns Hopkins page, as probably everybody else, so the lethality at the moment for the confirmed cases is 5.5%. But let's see, at the end of the days when the dust all settles, we might actually know how dangerous this virus was. You know, dangerous. It's super dangerous. We have to take this very serious. Well, the whole globe is shut down by this pandemic, so everybody of us is taking serious. But so this is not a nice virus because this has all the makings of a pandemic, how it spreads, that people can walk around being healthy, that it causes really very severe disease, and also that it actually hit an entry point like ACE2, you know, which is in the lungs, and that's the reason why people get this deep lung inflammation, this deep pneumonia. But ACE2 sits also in tissues like the kidney, in the heart, in the blood vessels, what we've been just showing in our paper, in other tissues, and it spreads in other tissues. And this is why people get this multi-organ failure. So COVID-19 is not just a lung disease. It's a multi-organ disease. People have cardiovascular failures or kidney disease and so on. It even can spread apparently into the brain. One of the early signs of the disease is people cannot smell anymore and have lose their taste. So this is not a good virus to have around.
Well, that's extremely helpful, and actually explaining about it being a multi-organ disease as well as the similarities and the differences to SARS. That's extremely helpful. You know, my background's in journalism, obviously, and I co-founded the Conversation Canada as a way to try to surface scholarly knowledge and distribute evidence-based research to the public, to wider audiences. And so I'm curious, you know, we're all bombarded and inundated with helpful public health information from the public health officers, as well as from the media. So what are you seeing? What do you think is not being said, or what's information that you would want people to know about coronavirus that we're not getting? Or what would you like to highlight that's there that may not be fully articulated?
I guess we all have the information. We're bombarded by information. It's probably the first, you know, big virus epidemic which goes global because of social media. You know, the first case in Uganda, and everybody knows it immediately. So that's of course also part of this whole equation we are living in now. You know, the fear of the virus, and of course we should fear it. But of course, you know, with media reporting everything, whole countries being shut down, also for good reasons, because it's very important to do this. Because the problem, of course, with this virus is we've never seen it. We've never encountered it as a population. We encounter every year the flu virus, so we have some reasonably herd immunities for flu, even if the virus mutates every year. But this is a totally new virus for our immune system, and so we have to adapt to this. And yeah, so, but I mean, I think the message is it's a virus. We can understand it. And because we can understand it, we can do something about it. So we have to take this virus very serious, because that's what we see. Unfortunately, many people have died. Many people have gotten very sick. But I'm also quite hopeful that that a lot of companies and academics, like our group, is actually advancing quite fast. And you know, if we actually look at the whole world now, it's quite an interesting experiment, because probably every university has been shut down. Most of our work has been shut down, and basically everybody is working on one topic. So that's probably the largest scientific exercise in our history. You know, every university on the planet and companies trying to figure out how this disease works or what we can do about this. And you know, some years ago, nobody cared about ACE2. Now it's probably the most researched molecule on the planet within 1 month. And so this is, I find, quite interesting. And also what I find interesting from my perspective as a researcher, how amazingly fast research has become and how amazing technologies have become. You know, we know this virus for 2 months, but we know unbelievable things about the virus. And you know, how it looks like, the structure, the 3D structure, every sugar on the virus, you know, which cells are being infected and so on. So I'm quite hopeful, because we can understand it and we will figure it out eventually.
That's a great segue to basically your therapy and treatment. So you're saying that because we understand viruses, that's a positive, and that's a way to sort of that we can make sense, that we are making sense of it already. So can you tell us a bit about the process where you are now in terms of treatment, and how long it's going to take, basically?
Yeah. So first, my apologies. I drink my tea, so I got the cough too and a little fever. So I hope I didn't get the virus. That's just springtime allergies. So where we are, because based on our old research, so we developed a medicine which looks like the gate the virus takes. So, and this medicine basically blocks the virus to enter us or enter cells, and at the same time protects tissues from getting damaged. So it has two functions engineered in one molecule. So we have been developing this actually for 15 years now. It has been tested in healthy volunteers and also in patients with lung disease. And if everything works well, this biotech company I co-founded many years ago in Europe will actually start a phase 2 clinical trial in Europe and hopefully soon also in China. So basically what we will do is we will treat 200 people with severe COVID-19 disease with this drug for 7 days. The first recruitment will maybe happen even tomorrow or by Friday, or latest early next week. There will be 10 trial centers in Europe, 2 in Germany, Munich, Hamburg, 4 trial centers in Austria, because this is where we come from, and actually the government gave us around $10 million to work with this and set up the clinical studies. So big thank you to them. So the university clinic and medical school in Vienna, Salzburg, and Innsbruck will participate, and actually 5 hospitals from the University of Copenhagen will participate. So what's very important, this trial will be a placebo-controlled, double-blinded trial. So nobody of us will know who actually gets the drug and who doesn't get the drug. The treatment will be 7 days, twice a day, intravenous infusions, and every patient will be followed for 3 weeks afterwards, what happens to them. The patient populations are cases of patients with COVID-19 which are quite severe. So they are not at the late stages, but more or less they're already quite sick. They need already some oxygen. And the idea would be that we clinically help them so they don't go into the late stages. And of course, hopefully we prevent people from dying. So this is the population and the cohorts we will treat. And it's very well controlled. And I would really like to say that to have controlled trials in this pandemic, in this chaos which is happening at the moment, is absolutely essential. You probably all have heard, and you have heard about chloroquine, right? The US president is claiming it's working. Others are claiming it's working. I had a call with people, some friends in New York 2 days ago. They're all taking chloroquine now. So this is actually based on the clinical study, which was not controlled and which does not allow us any conclusions. So of course we all hope it works, but the bottom line is we have no data that it's working. There's a trial in China which showed it does not work. So I really, it's very important that we despite all this mayhem and this chaos, and of course, many people not knowing what's happening, including the World Health Organization, claiming these things which we should test. But even in this chaos, we should, not put aside things which actually always helped us. Very science-driven clinical studies. Because at the end, we need to know, does it work or it doesn't work? Because nobody's helped with bad data and bad clinical trials. So that's why we made absolutely sure that we make a clean and controlled study. Because at the end of the day, we need to come up with a conclusion. It works, or which we all hope, or it does not work. But to have some conclusions based on some hope, conjectures, it's not very helpful.
Well, that's definitely hopeful that it's maybe starting tomorrow. Thanks for sharing that. That's good news. You mentioned that it's somewhat of a global experiment, unfortunately, with the obviously dire consequences happening on the ground, that there's all these scientific teams and scientists working on this around the globe. What are some of the other research in terms of treatment that's going on that you think is interesting or might be relevant in addition to yours?
So at the end, we have to get at the virus from many angles. This is what was the breakthrough in HIV therapy. It was not just one angle, but 3-4 angles. And then, then we had new medicines which really worked. So we're feeling that the single medicine will not be enough, that we have to combine it, and understanding the disease better, then we can actually combine it. And of course, one thing we need, and there are many people are working on, is of course a vaccine. The many approaches to this. At the end of the day, because we need safe vaccines, we cannot just immunize people and don't know what happens. So this, the earliest, if everything works well, this is from a year from now or 1.5 years from now. And then, of course, even if we have a safe vaccine, to vaccinate the planet will take quite a while, especially for a virus like this. So I read up a little. Up to now, I think we've only eliminated a single virus with vaccination on our planet, which was polio, which also took decades to actually do this. So we need a vaccine, but the people should not bring up their hopes. This will solve everything, and in half a year we've vaccinated everybody on the planet, because that's just not possible. But it's important, and we need it, and we need good vaccines. The second thing is, of course, what do we do when the virus already gets to us and there's no vaccination? How would we actually block the virus from growing? This is basically a part of our medicine. This is, for instance, what the famous Gilead medicine is doing, blocks the virus from growing. So this makes a lot of sense. Let's hope it's working. So basically, if there's an infection, it gets into the cells and it grows, and you can address this. We close the gate so it doesn't get into the cells, or if it's in the cell, you block the virus from growing. And of course, the third thing which we need to do, and many people are working on, is there's the severe inflammation. There's a cytokine storm, and this is actually what damages the tissues and the lung. So what can we actually do about this? How can we figure out the immune system? How can we trick the immune system? How can we calm down the immune system again? And of course, other systems in the body to not cause the severe disease, to not overreact to the infection. So at the end of the day, we have to come up with many approaches to fight this virus. So of course, I hope that our approach will work. We do careful clinical testing, so at the end of it, we know if it works or maybe it doesn't work, but there are many other approaches we need to go after, and of course combine approaches to really conquer this disease.
Yeah, that's helpful and that makes sense. So in terms of, obviously there's interdisciplinary research, global research teams, all of these partnerships already in the scientific community and in your research areas. Is the coronavirus, is it stimulating even more collaboration? Like, do you see the nature of scientific collaboration shifting at all in the midst of this?
It will, and it already did. At least in my community, everybody wants to work together because, it's a virus. You need the live virus. You need experts who actually can grow the virus, special laboratories where we can grow the virus in safe environments. We cannot just take the virus and put it in every laboratory environment, so this is not safe enough. So we need to collaborate anyway. In my case, I work with tissue engineers, with stem cell people, with people who do immunology. And when I realized around the last week of January that ACE2 is actually the receptor also for the new virus, then of course we realized that our work we had done 15 years ago becomes very relevant again, because it's a hard infection. And within a week I could put together a team of virologists, tissue engineers. Everybody immediately responded and wanted to be part of the team. So from my personal experience, this was actually quite amazing. So everybody stopped being competitive and was very happy to be part of this whole exercise and contribute. And what I also find, because we are in the heart of this whole storm, I get emails from people from Israel, from Iran, from from all over the planet, bringing in ideas, what we should do, how we could address things. It's unbelievable how, who works on it. Yesterday, a plant engineer from Iran emailed me, and and he said, "Oh, you work on ACE too." I had the same idea. Plant engineers are doing this, and it's amazing what has happened.
Yeah, it sounds like a truly global scale of collaboration. So you talked about, obviously this the coronavirus pandemic resurfaced your old research from 15 years ago. What were you doing? What were you researching? What were you focused on just before this hit?
Oh, the new virus. Oh, yeah, we actually got another paper accepted in Cell the same day. We got our other paper accepted on the virus. On the virus paper, we actually worked for 6 weeks, so it must have been a world record from starting of the work and get the paper accepted into Cell. Cell is one of these grand journals for us, Nature, Science, and Cell. For the other paper, we worked on for 10 years, just as a comparison. And it's actually a gene we found which controls thinness. So we asked the question, why can some people eat as much as they want and they still stay skinny? We all know these people who can, snack the whole day but don't gain any weight. And I think we found the first genetic insights on thinness. So we are not studying obesity, we study thinness. It was quite interesting. It's actually a gene in our head which functions in our head, and it controls for every calorie we eat, we burn much more if we turn this on. So anyway, so that's what I have been working on. And the other thing we have been working on is we developed the first human blood vessel organoids. So we did stem cell engineering. That's actually why we could now use our blood vessel organoids, tissues grown from human stem cells, which look like blood vessels, and we could use them in our study for the virus now. So that's what I do.
Great, well, thank you, and congratulations on your other publication after 10 years. It's a long time to work on one area.
It's not out yet, but soon. Not out. What's really bad, nobody cares about it anymore.
Exactly, everything's moved. Completely true. So I think it's time to move. We've got a ton of good questions on Slido. So if you're okay, Josef, do you mind if we move to some of the webinar participant questions?
Yes, please. Yeah.
Great. Perfect. So the questions move up, that people like them, and so these are the most popular questions that everyone in the webinar would like to see answered. So the first question is, what happens if the virus mutates? Do you anticipate this happening, and can the ACE treatment be modified to adapt?
Yes, so that's a very good question. For instance, when the people developed the first inhibitors for the HIV virus and also for other viruses, the virus has genetic material, and this has to grow and replicate itself. And these are some of the drugs which hit this pathway, so the virus cannot do this anymore. This is, for instance, what the Gilead drug is doing. But for instance, if we infect our cell cultures with around 1,000 viruses, we get around 1 million out within 15 hours. So the virus is growing like crazy, and of course they can mutate out of one of these pathways. So there's a fear that there will be a resistance of the virus if you only hit them at one weak spot. And this is exactly what happened for HIV, and that's why HIV treatment needed a combination of 3 drugs hitting at 3 different weak spots of the virus. So yes, probably the virus can mutate itself out of these drugs. We don't hope so. It could also mutate itself out from antibodies. If people develop, today there was an announcement of the big pharma which invested, I think, $300 million in monoclonal antibodies, which should neutralize the virus. So probably the virus can mutate itself out of this. But it cannot mutate itself out of ACE2 because it needs ACE2 to enter our cells. So it could mutate that it binds better or not as well, but at the end of the day, it still needs ACE2. So this is actually why host factors treatments which don't attack the virus directly, but actually treat our body, our cells, from protecting them from the virus, the virus has a really hard time to mutate out. So, of course, we cannot predict it, but we believe ACE2, the virus, will not be able to mutate out of ACE2 unless it finds another receptor, which we cannot predict, but it cannot escape ACE2 inhibition.
Great, thank you. Another top popular question about ACE inhibitor is, what advice is being given to patients on ACE inhibitor medications?
Oh, geez, that's a loaded question. I mean, the whole American Society of Cardiovascular Research and the European societies have basically said there's no reason to take people off of ACE inhibitors. So that's basically also, what I should say. If something, talk to your doctor. So the biology is the following. ACE, angiotensin converting enzyme, makes out of a peptide which is called angiotensin 1 to 10. It cleaves the last two amino acids off and makes angiotensin 8. And this is where ACE2 acts on. And angiotensin 8 acts on a receptor which is called the AT1 receptor. So the blood pressure medicines are basically blocking ACE. So angiotensin 2 is not made. The reason why this blocks blood pressure is because every time when we get now up after this talk, actually angiotensin 2 is released and it controls our blood pressure. So if you have a lot of angiotensin 2, our blood vessels become smaller, which means we have a higher pressure. So this is why ACE inhibitors block this, but they can also block that you don't make angiotensin 2, the ACE inhibitors, or you can block the receptor of angiotensin 2. These are the famous ARBs, angiotensin receptor blockers. And ACE2 is in the middle. It actually takes angiotensin 2 away. And so it does not bind to its receptor anymore. And it also makes good peptides. Something is called the angiotensin 1 to 7, 1 to 5. So the biology is actually a little different, because if we don't have Ang2 anymore because of the ACE inhibitor, then ACE2 can actually not work anymore. So, and it's different from the blocking the angiotensin receptor. So we have all these peptide studies, which are happy to share with everybody. So that's what the science tells us. And of course, there's a difference to the clinical practice. And again, I can only reiterate what the societies are saying, and the medical university is saying, talk to your doctor. And at the moment, we don't have the data which would, really tell us that ACE inhibitors would be bad. So that's actually, some people wrote editorials, take away ACE inhibitors because they're bad. But the bottom line is, we need real data to come up with this conclusion.
Great.
So
Thank
anyway,
you.
this was probably born on my research back in 2005, because we actually showed the lung protective function of the system is the enzymatic function. And when we block the angiotensin 1 receptor, we can actually block from lung disease also. So it's a double-sided sport.
Great, thank you. The next question is, why is it that COVID-19 is able to survive on surfaces while SARS requires direct infection?
Hard to tell. Probably the SARS virus also survives on surfaces. We just don't know. Was not really studied, or should be studied better. And also the data on the surfaces are very different. There are data out there where it says it survives for 7 days. Some other people claim it survives for 1 day. So clearly it does survive. It might depend on humidity, if it survives on certain fabrics where the virus can actually live on. So there are probably many conditions. What I found really interesting was a study from Singapore, where they looked at the room of patients, of COVID-19 patients, so they knew they were sick, and just to see where is the virus. If you have actually people who carry the virus and they're quarantined and sent home, and so where do you actually find the virus? And they actually found it all over the place, from the toilet seat to doorknobs to the chairs to the couches. So the virus was basically spread all over the place into the air conditioner and so on. So
okay, thank you. The next question is, do you have any thoughts on why only some people develop clinically significant COVID as opposed to others?
No, that's the $1 million question. I think nobody understands. So the disease in many people, they get little fever, cough, and then they recover. Some other people get more like flu-like symptoms with very high fever, some rashes on the body. They don't smell anymore, they cannot taste anymore. So the more severe disease, and then they recover, and some people all of a sudden the next day fall into this severe lung disease, like Boris Johnson we have seen now, have basically observed life in the media. So typical course of the disease, fever was still reasonably okay. So yes, this would be really, really important to understand why somebody develops the disease much worse and some others don't. Of course, what happens when the severe disease happens, so the severe disease and then of course the multi-organ failures at the end, is that the immune system just overshoots. So somehow our immune system is hyperacting against the virus, and then this causes then the blood vessels to leak in the lung, the fluid goes into the lungs, and of course damages other tissues. So this it's something called a cytokine storm. So this we really need to understand why some people get it and some don't. I have no idea why. And I talked to some people who really understand immune systems and immunity against virus infections and how you design vaccines. Some really of the world leading people on human immune systems. And so just a friend of mine in Stanford, I talked to him, they have also no idea what's really going on. So there's a lot we don't understand in terms of our immune system, how it reacts to the virus. And this will be a critical question.
Yeah, and you can see it in the media coverage of people who are otherwise healthy. I think a lot of people are asking that question, definitely. Another top question is, will coronavirus, will it disappear like SARS, or is it a long-term disease, do you think?
So I can, I'm not an epidemiologist, so this is not my field. So I can just basically say what I hear. So one thing which could happen is, our measures which we put in are working, and then people get sick, and there will be of course many more people get sick, and summer comes and the virus disappears. So this would be of course the perfect scenario for everybody. The chances for this to happen is probably below 10%. So what probably might happen is that the virus becomes seasonal, like the flu. So now we, with all the measures of people, we have to stay at home and travel is restricted, and basically public life is more or less shut down. The curves will slow down in summer. With warmer climate, it probably will disappear. But of course, we have to open our countries again. We have to open our economies again. By the way, Austria, where I am, we will be the first country which opens the country again by next week. So the small shops will open again. So it will be interesting to see. But the epidemiology tells us that it will slow down. The virus will probably go to the southern hemisphere. But by November and December, around Christmas, there will be the next outbreak. So we will have to be prepared that this is a virus which we have to live with for many, many years. Even if there are vaccines, there will be always outbreaks, something probably like the flu. And but of course, after this first outbreak, we will know the virus better. We will have probably first treatments which work. And so I think this what happened now on the planet will be a unique event, because the next time the outbreak happens, we will have more prepared healthcare systems. We will have medicines, hopefully, which work better to keep people out from the severe and very severe diseases. So I think what happens now will be a unique event, but we might have a future where we have to live with the virus for a long time.
Great, thank you. That's very helpful to know. Yeah.
I hope I'm wrong, and the first scenario is true, and it just disappears. So the problem was the problem with infections, Ebola, as bad as it is, if Ebola would break out in Canada, only people are infectious who have the disease. So we would probably, as terrible as it is for the individual, with a public healthcare system like in Canada, we could contain it reasonably fast. So this is what happened with the first SARS outbreak, where you could follow every single patient, where they got infected. And of course, if you can do this, you can contain things. But with this virus, we just don't know who has the disease, who carries the virus and is walking around healthy. So the future will be also probably, that we will have to test much more. If I may, allow myself a little critic on Canada, as they were not really prepared for testing, because testing is critical. We have to protect the healthcare workers. We have to protect people who take care of older people of the community. We have to know how prevalent the virus is in our communities and do something about this. So the future will be, we will reopen our countries. We have to. There's no other choice. We'll probably have to live with the virus and have a new normality, where we go in restaurants and clean our hands and walk maybe with masks around if we feel a little cough ourselves, to not protect ourselves, but to protect the others from ourselves, and have testing stations. Actually, somebody talked to me today, ideas combining computer chips and how they do them with testing. So, in the future, there might be tests where you go to a restaurant, you get a little swab, and in 10 seconds, you might know if you carry the virus or not. So that's probably one of the futures we have.
Thank you, because there's been a lot of social media back and forth on Canadian Twitter, basically about, is Canada testing enough? And there hasn't been a sense of what's the data that we use to try to assess that. So what would be enough in Canada? How do we what's the comparative, or what's the proportionate? What's the number?
So I don't know should be the numbers. I just know from South Korea they're testing around 20,000 people a day, probably even more. So the system there works. Everybody can get tested if they want. If you get tested, you pay, I think, $100 or $150. If you're positive, then the healthcare system pays for this. So, and this basically allows everybody of us to actually go be tested if we want to. What I understand from some of my friends. At the moment, again, it's no critique. It's very difficult to get all the reagents and testing to set up proper testing. But what I hear from some people in Canada is, they have a cough, they actually want to get tested. They're told you don't qualify for being tested. A friend of mine who actually told me she was sick for 2 weeks, was always rejected to be tested. And 3 days ago, somebody who cleans her apartment called that she was tested positive. So I think one could probably do better. Let's put it this way. We all have to. So even the Austrians, we have to. Also, this is actually a country, because I'm actually advising the Austrian Prime Minister and the government. So I think they did a really good job here, how to communicate it, shut down very fast, react very fast, because it's really tricky. How do you communicate to people? They have to close their shops, right? People lose their livelihoods. How do you communicate to people to close their restaurants? So what's happening in the world now is unprecedented, and of course, all the communication for this is very tricky.
Mm-hmm. How long has Austria been shut down for? When did you close?
We've actually also, like every other country, in Italy, there were already the outbreak, so we knew. In Austria, they were still happily skiing. The ski resorts is high season. Of course, they didn't want to close the ski resorts. What happened was, and you probably followed all the story, is in the ski resorts, some people, like barkeepers, were infected. And of course, being after skiing in the après ski, it's the perfect incubator for a virus. It's loud. People talk loud. So probably half of Northern Europe was infected by skiing in Austria. So that's one of the estimates. Because basically the first cohorts of people in Norway, they all went skiing in the Alps. So this is actually what happened. So, they also were not prepared. Let's put it that way. And everybody is, of course, hopeful. It will not affect us. Also, we already knew it started in northern Italy. Switzerland was heavily affected because they have 200,000 people coming over the border every day from Italy to work in Switzerland. Of course, Austria is just next door to Italy. So it was just a matter of days for us to happen. Considering this, we actually still reasonably okay compared to Italy, which is a total disaster. And what I also learned, which I find really interesting, there were global, global warplanes. What would actually happen is if a pandemic hits the world, and northern Italy was always the first place which would be the epicenter of an outbreak in Europe. So people knew that this is actually the place where Europe will be hit first. The reason is, you might know.
I don't know.
Any guess? The fashion industry.
Oh.
Because it's so much connected all over the planet. In northern Italy, there are, I think, 350,000 Chinese working, in the fashion industry. And so this is how it spread into Europe.
Oh, I didn't know that.
Yeah, and this is actually where it happened, because fashion, of course, is very well, it's global, there are global contacts, and so this is how it got in. And exactly the Italian places where it started, around Bergamo, these are the major places for the fashion industry in Europe. And then, of course, there were soccer games. There was this famous soccer game, the Champions League. I like soccer, you probably know. So there was a soccer game of Atalanta Bergamo in the European Champions League playing against Valencia. And the game was in Milan. And I think it was 50,000 people watching. And this was one of the major incubators of the virus in Europe, because I think half of the Valencia soccer team got infected on this day and so on.
Great, thank you. And I didn't know that about
traveling all over Europe and moving around. And that's why they actually had to shut down all this, all the soccer games with spectators very, very fast, because to kind of control the spread of the virus.
Great, thank you. I didn't know that about the fashion industry. Last question from Slido. There's talk of other treatments, HIV drugs as being possible, plausible. What are your thoughts on that in terms of other possibilities?
So yes, so the various drugs people have been promoting, especially the World Health Organization. So the World Health Organization, they're doing something called the Solidarity Trial, where they basically proposed 4 treatments endorsed by the WHO. One is chloroquine and hydroxychloroquine, which I find, forgive me, we need many more data that, to know if this actually works. So actually, if I may also have a little critic again, I have no idea how the WHO can support this. Honestly, they should support things based on hardcore science, not on some, guesses that something could work first. The second one is the drug of Gilead, remdesivir. So these are now my personal opinions, not official opinions of anyway. So this actually, the Gilead drug makes sense because it gets at the virus where it grows. The HIV drugs, this is a drug which hits the virus at another weak spot of it, which has initially been developed for HIV. So it does make sense to be tested. There were already 2 clinical trials. It's not working. So I and many others don't have too much hope on this. The Gilead drug also was not developed for this new virus. It was actually developed to treating Ebola infection. So at very high dose it works, but it's not a very specific drug either. And there's a combination with interferon. So this is what they're doing now, what they're promoting, supporting. You of course know what's going on south the border. People who support science and the scientific method, like Tony Fauci being attacked, that he doesn't know what he's doing, and so these things have become and these treatments have become very emotional. I can understand, we don't know how to treat these patients. Because everybody's looking for some hope, for something which we could use. one can repurpose this drug. So I can understand why this is supported, but I think it's very important to have solid data if they work or not, because nobody's helped by doing clinical trials on patients which actually might not work. And because what this is also doing, it takes away or it blocks possible clinical trials on drugs which actually make much more sense. So I think the WHO would be much better advised, and I have nothing to do with them. Would be advised to support, drugs which are based on fundamental scientific principles.
Thank you, Josef. And we're actually running out of time. We've probably got, do you have a last minute comment before we go to the polling question and I say thank you to you and everybody for joining us today?
Yeah, last minute. So it's a virus, we can understand it, we will understand it. By summer, we might have first drugs which work, and then I think we have a much better handle and the world will come down again. So it's a unique time, but we will live through it. And of course, science at UBC, which is a great university, it's my university, will be the solution, and we will contribute to solve this puzzle and save lives.
Great. Well, thank you, Josef, for joining us today. That was extremely enlightening and helpful to know that we can understand it, and that it may be around for a long time, or it's likely to be around for a long time, although that's not what you would wish. So thank you for all of the clarifying questions and comments, and for all of your excellent research on behalf of Canadians in the globe. And thank you to our viewers, UBC alumni and friends, and for everyone for joining us for today's important conversation. The video from this webinar will be available later today at alumni.ubc.ca/COVID-webinar. There you can also find out how to support UBC student emergency funds at this time. Before you go, we have a few minutes left to answer a brief question, yes or no, in terms of, did you find this webinar informative and useful? Please answer yes or no. It will work as we're here. I'm giving it 30 seconds. I wasn't told if there was a bell or something to let me know when 30 seconds ended. So there, it's over. Thank you so much for your feedback. The poll is now closed, and we wish everyone well in this time of uncertainty and new realities. So stay home, be safe, and stay healthy, and have a great rest of your day. And thank you again, Dr. Penninger. It's been a pleasure and an honor.
Yeah, all the best to everybody in Vancouver, and I hope I can join you soon when the borders come back and the airplanes fly again.
Yes, well, we'll look forward to you coming back.